The first question, or the first thing you might ask, is why are we interested in computational protein design? Well, fundamentally, we're interested in understanding what the underlying physical principles are that govern the folding, stability, and function of proteins. Typically, or historically, to address these sort of relationships, we've used perturbation-based approaches. So, for example, if you're interested in understanding how an enzyme works, a protein, you might make mutations, perturbations in the active site of the enzyme, and then assess experimentally what those perturbations did to the function of the protein. What we're after ultimately is using a design-based paradigm to study these relationships, folding, stability, and function. And in a design-based paradigm, what we're trying to do is to first construct a mathematical model, computational model, of all the things that we believe are true about proteins, how they fold, how they achieve stability, how they function. And then within that computational construct framework, we want to then run a design calculation, that is, design new or novel proteins, and ask the question experimentally, are those proteins viable? Do they fold? Are they stable? Do they have function? And if they have all these properties, then we can conclude that our computational model, our mathematical model, actually accurately captures the fundamental essence of what a protein is. On the other hand, if the experiments don't work, then hopefully we can learn from those failures to modify our model in a way that allows us to go forward in terms of building new knowledge into the model as we do designs and experimental validation. As a chemist, ultimately, I'm interested in designing new systems, engineering new systems, and in particular, we're interested in things like engineered antibodies, biosensors, industrial-grade enzymes, research reagents, et cetera, all the way to using these sort of methods to design human therapy. Okay, so before I start then in terms of protein design in particular, let me just give you a brief overview of biology. So everything you need to know about biology is actually on this slide. Importantly, we start with DNA. And as you know, DNA gets transcribed into RNA, and then RNA gets translated into protein. We're going to focus on the protein end of the spectrum. A lot of the hype recently has been in genomics, that is studies related to DNA, but the real action ultimately is at the protein level because proteins are the things that actually do stuff in cells. They're the molecular machines. They're involved in signal transduction and metabolism, in replication, in essentially all aspects of cellular function. So in general, proteins are linear polymers of amino acids. The structure of the protein and oftentimes the dynamical behavior of the protein dictate the function of the molecule. This is important because we use this idea of the relationship between structure and function in our design efforts. And as I said earlier, proteins are involved in essentially all cellular processes. Across the bottom, we have examples of 3 different proteins, just sort of randomly selected. On the left side is a protein, a small bacterial protein called protein G, and it's shown in full atom representation. It's about 60 amino acids, and you can see it's a fairly complex structure, even though it's a relatively small protein. In the middle is a protein of roughly the size of 300 amino acids, shown in a ribbon diagram. And this protein is an enzyme, and you can see if you look closely near the middle of the molecule, there will be a small molecule substrate. This is around, as I said, 300 amino acids. On the right side of the slide is a membrane-bound protein. It's all helical, and it resides in the membrane. The membrane would be, form a layer like this, and this thing would stick in the middle. And you can also see that in the middle of that protein, there's a chromophore that absorbs light and is part of the function of the molecule. Okay, so when I talk about protein design, I often get questions that are related to protein folding. And so let me take a minute to distinguish between protein folding, and that is protein fold prediction, and protein design, which is often referred to as inverse folding. Okay, so in protein fold prediction, we have an amino acid sequence the question is, can we make a prediction about the structure that the protein will fold to in solution? Now, we can think about this in terms of the diagram on the bottom. We have sequence space. Sequence space is large, and with that blue dot in the sequence space diagram is a representation of the amino acid sequence of interest. The mapping then that we're interested in is going from sequence space to structure space. And this is a difficult mapping. One reason is that you're either correct or incorrect in the sense of there's a single correct structure that this particular sequence will fold into. On the other hand, protein design is essentially exactly the opposite problem. We start with some structure that we're interested in, and the question that we're asking is, can we predict, in our case compute, an amino acid sequence which will fold to that structure if we put it in solution in the laboratory? And so the mapping then is exactly the opposite. We're going from a point in structure space to a set of sequences in sequence space. Now you'll notice that in the inverse direction, there will be many solutions in sequence space that satisfy our target structure. And this degeneracy, that is this one-to-many mapping, aids us in the protein design process. So protein design fundamentally is a much more straightforward endeavor than protein fold prediction, because of the idea that there are many solutions which actually lead to the correct answer, that is, the correct structure in solution. Library design, protein library design, is related. So in this case, rather than trying to identify some single optimal sequence for our target structure, we're going to, again, use the same mapping procedure, but we're going to now specify an entire list of sequences, all of which are predicted to fold to the correct structure. And then in the laboratory, we can actually take this list of sequences, the sequence alignment, and manifest it as a nucleic acid library, as a protein library, and then screen or run a selection on that library to find those examples which actually satisfy our design objective, folding to the correct structure and ultimately having some desired functional characteristic.